cardiometabolism. And today, I believe the talk is going to be on sort of microRNAs that are maybe not exactly as we expect them to be, which is fabulous. And I think the understanding of, Christian's also contributed so much to the understanding to microRNAs and atherosclerosis and cardiovascular disease too. So this is going to be a very interesting talk. So welcome, Christian, and thank you very much for your time. And usually at the end, we have sort of a Q&A and people just kind of raise their hands. So we'll coordinate that at the end. So take it away.
Yeah. Great. Well, thanks, Katie, for the kind introduction. I'll try to release here my screen. Yeah, it all seems to be working. Yeah, and thanks to Katie and Ibro and Esther also for this great initiative to have this very cozy format during these difficult times. And I think this really contributes a great deal to keeping up with science in these times. Well, so now I have my laser pointer, and as alluded to already, I wanted to present some very unusual data, some of which has been published last year on non-canonical functions of microRNAs in atherosclerosis, and this is also intending to challenge some of the long-standing dogmas in microRNA biology. I just have to put my screen a little bit smaller here. Okay. Yeah. So as you all may be familiar with, the biogenesis and canonical functions of microRNAs has been well established. So microRNAs are processed from premeres by the Drosha microprocessor and then by DICER into a mature hairpin structure, then into a microRNA duplex, and this can also then be exported into the cytoplasm. And so basically some of the more long-standing dogmas were basically that the passenger strand is typically degraded and is non or less functional. And then the guide strand here is loaded into RNA-induced silencing complex through binding AGO2, so Argonaute 2. And basically thereby microRNAs can interact with target RNAs in this RISC complex to promote either the decay or translational repression through multiple mechanisms. So I would like to start basically with this first dogma, and we have a sort of prototypic microRNA, which is our pet in the lab, which is microRNA 126, which is one of the most abundantly expressed microRNA in endothelial cells in the EGF7 locus. And you can appreciate here that the expression of a guide and passenger strand that can be observed for 126 between the 3p and 5p strand can be balanced. So other than in many other examples, for instance, LED7A and 121, where the 5p strand is more like the predominant guide strand, or 22 and 29A, where it's really much expression of the 3p strand. The expression of 126 strands seems to be balanced. And so this seems to indicate that there are specific mechanisms that control the stability and homeostasis after basically expression, which is obviously different to other microRNAs. In addition, the two members of the microRNA 126 duplex have been shown to be functional by repressing distinct targets and providing complementary function and functionality in a dual system for endothelial protection. So for once, the 126 3p strand can be delivered by apoptotic bodies into endothelial cells, and thereby limits atherosclerosis by inducing angiogenic recruitment, by promoting the expression of CXCL12 through repression here of RGS16, a negative regulator of G protein coupled signaling. But it can also have, I have to use the laser pointer a little bit better, anti-inflammatory effects that are also important in atheroprotection through repressing VCAM1. In contrast, 126 5p, the partner strand, basically is depleted at predilection sites of atherosclerosis, in particular when you come in with a second hit of a high-fat diet. And so basically the knockout shows a massive exacerbation of atherosclerosis, so clearly indicating that it's atheroprotective. And if you add basically mimics, you can restore the endothelial proliferative reserve. This is shown here, in particular in these low shear flow areas, and thereby mediate atheroprotection. And this is yet again mediated by repressing a completely different target, the Notch inhibitor DLK1. This we took as a starting point, and indeed we could see that this high shear stress basically can, in a KLF2 mediated process, induce a preferential upregulation of the 5' strand. So this is shown here, and this was both basically true in vitro, but also in vivo, where you can differentially analyze low or disturbed from normal high laminar shear flow areas. And this could also be observed by KLF2 overexpression here in endothelial cells, where you could see a preferential induction of the 5' strand, a bit following the pre-microRNA, but not the 3' strand. Yeah? And likewise, in the KLF2 endothelial-specific knockout, only the 5' strand was affected. So together, if you assume again that basically the 3' and 5' strands are derived from the same pre-expression, this clearly indicates actually a post-transcriptional mechanism to explain these differences. Yeah? And from there, we basically went on to look what KLF2 is also doing in cellular homeostasis, and there were interesting papers out there implicating KLF2 also in autophagy programs and autophagic flux in endothelial cells. And indeed, we could show that high shear stress and KLF2 actually upregulate autophagy-related genes, most notably ATG, so ATG5 and 7. I'll come back to that later. But also the overexpression of KLF2 induces autophagic flux, as shown here by the typical LC3 2-to-1 ratio. And so basically with this, I'll come back to this later, because this is going to be an interesting mechanism of how this homeostasis works. But coming to the second dogma, basically the risk complex that I alluded to is actually encompassing a very heterogeneous family. So basically AGO2, which is the eponymous protein that defines the risk complex and that binds the microRNA, can recruit several other proteins, including TNRC6A, to facilitate this gene silencing, and this is typically then observed in a high molecular weight complex. So if you look at this gel filtration. And interestingly, mTOR, so remember mTOR inhibition is an important stimulus of autophagy. mTOR activation promotes the assembly of these high molecular weight complexes. Basically, this is quite interesting because also unlike in cell lines, where you can see basically here this high molecular weight complex, in vivo, AGO2 bound microRNAs exist mostly in a pool of heterogeneous low molecular weight complexes. And so we thought that the functional relevance of this was really not very well addressed, and basically there may well be non-canonical interactions of microRNAs within these low molecular weight complexes outside the typical risk, and also maybe exerting some post-translational effects on protein activity outside the risk. So mTOR inhibition with rapamycin, remember, would also induce autophagy, actually favors the occurrence of these low molecular weight complexes. So you can see basically also the shift in this interaction basically of TNRC6A with AGO2 basically is lost basically when you induce here with rapamycin, and you clearly see a shift towards more low molecular weight complexes with less sort of co-elution of AGO2 and TNRC6A. So basically, we thought this may also be part of the explanation for this differential effect on this two strands of 126. And indeed, mTOR inhibition here with rapamycin specifically preserves the 126.5p levels in endothelial cells, and this is in a process that is dependent on autophagy, because you can see here 3p is degraded, 5p is preserved, and basically this is reverted, this effect with a knockdown of ATG5 and 7, or with a inhibitor baphalomycin of the late fusion of the autophagolysosome. Yeah. And so how does this work? How does this happen? Actually, so we used fluorescently labeled microRNAs 3p and 5p and stimulated with rapamycin to observe where basically the strands are ending up. And you see that clearly the 5p actually ends up sort of in a very nuclear location, so in a nuclear reservoir, while the 3p strand is more found in the plebs closer to the plasma membranes, or almost like being sorted for export in, for instance, microparticles or apoptotic bodies. And so the stimulation of cells with rapamycin clearly induced the nuclear copy number here of the 126.5p, not 3p, so very specifically. And again, this was a process that obviously required autophagy because you could revert it with ATG5 and 7 knockdown. And also it involved an active nuclear import, as shown here with the inhibitor ivermectin. So we then thought to look at this in more detail. So we used co-immunofluorescence staining here, and we also observed that rapamycin induced a co-localization of AGO2 with LC3, sort of, and this autophagolysosomal marker in the nucleus. And basically, this is seen here in the inset very well, and also co-IP showed basically that you have nuclear enrichment of AGO2, and again that seemed to be specifically carrying 5p and not 3p if you looked at the strand copy numbers. For further dissecting the location, we used a high resolution STAT microscopy, and this clearly revealed that basically this AGO2, because we were wondering, and some reviewers were also wondering, why isn't AGO2 then not degraded if it's in association with this autophagolysosomes? And the explanation for this is basically because it's on the it's sorted on the external membrane, and here you see the LC3 of the autophagolysosome, and it obviously is protected from degradation because it's actually on the outside. And this could also be confirmed here by trypsin protease degradation assays, which could actually show that basically AGO2 is preserved from degradation by being outside. Interestingly, here already MEX3A comes up, and I come back to that later because that's the protein that explains much of that. Okay, so we then went further on and also did some nuclear RNA-seq. So this is also was not published yet because we didn't have it available at the time. But we thought it's very comforting to see basically that with rapamycin you get basically not much, but one microRNA strand that is specifically picked up in the nuclear microRNA sac, and that turned out to be 5p. So in essence here, a completely unbiased approach that gives you basically the indication that this is a highly specific effect for 126 5p. We then previously had already validated that by qPCR for selected strand, and that they basically actually gave the same result with the specificity for nuclear 5p. Then we were interested to see what basically is the motives that are required for this nuclear translocation, because we found it also with AGO2 in the nucleus. So basically, AGO2 is basically binding the seed sequence of the microRNA, and matchingly and interestingly, we found that the seed sequence is actually not required for the nuclear translocation, because you still see it with the mutations in the seed. But if you go further here upstream here and mutate the middle motif in the microRNA, you lose basically the expression in the nucleus. So it seemed that this motif was more important for the nuclear transfer. We then went on to analyze specifically interacting proteins with AGO2 by co-IP, and so of the several known candidates basically that are known to interact with AGO2, there was nothing much coming up, but fairly new and recently described RNA binding protein MEX3A, that was the only one that basically was coming up in this sort of low molecular weight complex with AGO2. We then used basically confocal immunofluorescence to show basically that is also in the nucleus under rapamycin autophagy stimulated conditions interacting in the nucleus with AGO2, and also it seemed basically to be in the very proximity and presence of 5p, and this was then also confirmed by surface plasmon resonance here on an AGO2 chip, where you can clearly see that the interaction of MEX3A with AGO2 in a ternary complex actually requires the presence of the 126 5p of the strand. So you can envision that the complex forms through the microRNA with the two proteins, probably and likely bound, you know, at different motifs of the microRNA. And then basically we further followed up on that, also showing that in the MEX3A IP, we found a specific interaction with 5p. This was also followed up with a molecular dynamics simulation of the RNA binding KH1 and 2 domains of MEX3A that provided a model that both are actually required for binding of the microRNA. And eventually this was also confirmed by HSQC NMR spectra, where you could clearly see a shift basically in MEX3A in the KH domains upon titration of microRNA 126 5p. So basically, if you wouldn't have an interaction, you would see a perfect overlay of the red and the blue, but here you see these shifts. And here, SEX, so single X-ray diffractation actually showed that there is a compact conformation of the KH domains that is assumed upon addition of the 126 5p, while without it's basically more floppy. So basically you can envision that the binding of the microRNA actually brings these two KH domains together, and then it assumes a more compact conformation. And also this again for 5p specific would explain the differences in the experimentally measured affinities, which are always highest and higher for the 5p strand. Well, how does this work? And again, basically, obviously, this is important that this MEX3A RNA binding protein is required for the nuclear transfer of this ternary complex, because with MEX3A knockdown, you completely lose both AGO2 and also basically the microRNA in the nuclear complex. And again, this is the reverse nuclear RNA seq experiment with the MEX3A knockdown. There's one microRNA basically that goes down and specifically goes down. And again, this is basically 126.5p. We also have obtained some preliminary data indicating that MEX3A also interferes with the classical risk targeting cleavage function. So basically, it may sort of the interaction of MEX3A with the AGO2 complex actually may confer a less classical risk complex that is then basically more mobile and is more prone to nuclear translocation. And this already indicated that, you know, there are non-canonical functions of this nuclear 126.5p. What would it do in the nucleus? That there should probably be a functional relevance to it. And to test this and to address this, we basically devised a model where we basically mimicked the nuclear 5p presence by inducing autophagy conditions and then adding in addition 126.5p mimic. So under these conditions, you obtain the highest levels of nuclear 5p. And then we performed gene expression profiling associated with this nuclear 5p, and the key pathway enrichment analysis actually identified a predominant regulation of apoptosis-related pathways, interestingly. And indeed, when we used rapamycin and this 5p mimic in these conditions, we observed the inhibition of effector caspases, most notably caspase 3 here by the 5p. We then thought, okay, interesting, is this a transcriptional effect or not? But interestingly, these effects were already observed on apoptosis after 3 hours, so way before any transcriptional changes occurred. And again, you know, they were dependent on MEX3A, so MEX3A knockdown actually reverted these effects. So it indicated that there is some direct non-transcriptional interaction that is responsible for this effect. And then we thought, because we already observed that the caspase 3 activity is inhibited, maybe there is an interaction with nuclear caspase 3, which by definition is active with the 126.5p, and this was initially observed here with fluorescent in situ hybridization experiment, where you can see a very close localization, and you can also do a line scan here of these images of caspase 3 with 126.5p. And again, also we performed an IP of caspase 3, and again in the nucleus found an interaction with 5p and not 3p under rapamycin conditions. So indicating that actually 5p directly interacts with nuclear caspase 3, and again we performed SPR experiments here with the strands immobilized on the chip, and you can see basically that there's a dose-dependent interaction with caspase 3, so in the low micromolar to nanomolar affinity level, and no interaction with 3p. Again, NMR indicated that there's also a shift, so indicative of an interaction of the 5p with caspase 3. So what about AGO2? You were thinking, AGO2 is actually the strongest binder of any microRNA, but obviously here when we performed caspase-3 and AGO2 copies, AGO2 was not required for the binding or was not interacting here with cleaved caspase-3. And as a matter of fact, there was in SPR a competition for 126 binding. So if you add in here caspase-3 for AGO2 binding. So it seems that like there are conditions in the nucleus that favor a dissociation of 5P from AGO2, and then basically it becomes available to bind caspase-3. And this is also shown here if you analyze specifically the AGO2 unbound microRNA 126.5p. You can see basically a difference here under conditions with rapamycin. So you get a sort of this is due to the scale, basically a reduction of that, and that seems to be conferred here with MAX3A and caspase-3 IP basically to these fractions. And indeed, when you perform again the MAX3 knockdown and then analyzed in great detail the proximity between 5P and caspase-3, you see basically that you get that you lose basically the distance between basically and in this colocalization. So it seems also that this interaction or this dislodgement of 5P from AGO2 and binding to caspase-3 is favored by the presence of MAX3A. Okay, then basically what does this do? Functionally, basically it does, obviously again by SPR, caspase-3 is basically composed of two subunits which come together here to form the substrate binding pocket and an active catalytic center. And so 5P, if you titrate it into an SPR experiment, seems to inhibit the caspase-3 dimerization at this interface and thereby preventing the formation of the active center. And indeed, if you perform caspase-3 activity assays, you see an inhibition of caspase activity with 5P. Again, interestingly, not with the mutant that we used before, and at an affinity level or at a Ki that is basically remarkably low and comparable to the best commercially available inhibitors of caspase-3. So again, this basically was in difference to the transport, seed dependent. So basically, again, indicating that it's the sequence that is dislodged from AGO2 that is then also required for the inhibition of the caspase-3 dimerization. These are more specificity controls. So again, basically binding free energy. So computational molecular dynamics simulation indicated that the affinity was by far the highest for the 126 5P strand. Also, again, here caspase-3 IP indicated only 126 5P. None of the other microRNAs we see only with 5P specific interaction in surface plasmon resonance, and also this activity assay is only really specific for 5P 126. So what about, and this comes to the final slides, about the in vivo relevance. Basically, we generated a system with an endothelial-specific deficiency in autophagy using the ATG5 endothelial-specific knockouts. And in these, basically, we see that you lose the nuclear AGO2 localization. You lose significantly the nuclear 126 5P translocation, and instead you get an increase of circulating endothelial apoptotic bodies, so indicating that there is obviously endothelial apoptosis ongoing. And importantly, when these mice were backcrossed into ApoE deficient mice, we see, as we would expect, an exacerbation of atherosclerosis. And interestingly, this was most notable for the high shear stress areas, so exactly where we would envision the high shear stress, autophagy, nuclear import pathway to occur. As a follow-up, also basically we generated mice deficient in MEX3A. So at the time of the paper, these mice were not available in an atherogenic context yet, but we could observe here using fish in aorta en face, basically that also the MEX3A knockout reduced the nuclear 126.5p import in endothelial cells, and in turn increased active caspase-3 in the nuclei. So clearly indicating that the lesion of MEX3A favors endothelial apoptosis related to its effects on caspase-3 activity. And finally, to bring this also to the human scenario, we used 3D deconvolution microscopy here to show actually again in dissecting between high shear stress and low shear stress areas that nuclear AGO2 and 126 basically were higher in the high shear stress area, and the nuclear active caspase-3 was reduced. So what have we done further? Basically, this is also one interesting slide again that shows you that this is also again with high resolution microscopy that you also have an increased nuclear MEX3 125p interaction in arterial areas with high shear stress here. This is basically showing the fractions of interaction and non-interaction between the two in these areas. So clearly also indicating that the complex is there and promoted by protective shear stress. And so finally, this is unpublished and new data. We were able to obtain the MEX3A deficient mice and an ApoE background, and yeah, much to our delight, actually atherosclerosis is significantly exacerbated in the MEX3 deficient mice. And to further complement this also, and the relevance maybe to target MEX3A, for instance, by overexpression, we also used here in cooperation with Harry Schunkert from the Cardiogram plus C4D consortium and together with the UK Biobank, an conglomerate of different MEX3A SNPs that each single one, so these are not significance values that you expect to see from a GWAS analysis, but each of these SNPs individually actually is associated with increased cardiovascular disease risk in humans. And we are not exactly sure whether it's the numbers or why this has not been picked up in GWAS as of yet, but it's clearly an association there. Also here, if you look at the EQTL, you see an inverse expression association of this MEX3A expression with cardiovascular risk, and this is just shown here for one typical example of this SNP. So we clearly believe that there is an association, and it would be important and valuable to further follow up on that. So I come to my conclusions with this. So we've seen these several dogmas that we have challenged, and our data basically indicate that high shear stress and KLF2-driven autophagy can promote the nuclear translocation specifically of microRNA 126.5p to preserve and protect its levels. And this nuclear transfer requires MEX3A, which preferentially interacts with 126.5p in a low molecular weight complex with AGO2. And when dissociating from AGO2 in the nucleus, 126,5P mediates rapid anti-apoptotic effects by direct binding and inhibition of caspase-3. And this autophagy-driven MEX3 126,5P shuttle confers endothelial protection in vivo, limits atherosclerosis. The inhibition of caspase-3 by nuclear microRNA is a completely new paradigm that epitomizes non-canonical microRNA functions modulating protein and enzyme activity beyond any risk-related effects. There have been some other related examples on riboregulation, where there's also obviously risk-independent effects. So there's a growing evidence that there's much more to microRNAs than what's happening in the risk. And emulating these pathways, for instance, via MEX3A or interfering with overexpressing MEX3A may unfold multiple options for therapeutic targeting in vascular disease, and in particular in the apoptotic component in endothelial cells in vascular disease. And with this, I thank you for your attention. I hope I wasn't going too long, and we take, I always show this, we take great pride in our slow science. It takes very long, we are sometimes not publishing in the best journals, but we are very happy, to at least have and try to have a sound story and solid story in the end. Thank you very much.
Wonderful. Thank you, Christian. That was a really compelling and very interesting story. And I will ask the first question because I'm very curious for the MEX3, right? Is that the
MEX3A?
MEX3A. Does it do anything else? I mean, or does it need 126,5P to do it? Like, do you see what I don't know if I make myself clear. Like, so in the absence of, say, mere 126,5P, or in another cell type, or in other conditions, is MEX3A doing different things, or is it really there to mediate this dependent function on apoptosis and autophagy?
No, this is a very good question. So basically, it seems that it is an RNA binding protein, and so microRNA 126,5P is not the only RNA that it binds, and it has also not sort of primarily been described and found for its interaction with 126,5P. So I think under the specific conditions that we study in endothelial cells, we are pretty confident that this is basically the primary function of MEX3A. And I think, it's if you envision any protein that upon interacting with AGO2 could convince AGO2 to give up on its microRNA, this is a very strong and important effect. But, and I think you're right, and this is something that we're also looking at, there is expression of MEX3A in other cell types, for instance, in lymphocytes. And there we believe that probably the functional repertoire is a completely different one. And of course, that we are also generating the floxed mice. That's something that needs to be addressed.
Wonderful. Okay, Mabruka, go ahead.
Yeah, that's really very exciting science. Thank you so much for sharing this with us. So I'm going to naively ask you this question. So you elegantly showed that MEX3A could bind through KH1 and KH2 with mere 126. So how AGO2 could bind? Does it directly bind to MEX3A, or is it intermediate between?
That's a very that's a very interesting question, and that gets very deep into what we are currently doing. Because there was another ring domain. I can go back and open the presentation again, but for instance, in an NMR experiment, it makes a difference, in terms of obtaining evidence for a ternary complex, whether you just use the duplex of the KH domains or whether you use the full length MEX3A. Yeah. So we believe that the RNA binding is basically by this cooperative interaction between the two KH domains, but then the further interaction with the AGO2 complex is mediated by the other domain.
That's very interesting. Thank you.
Abru, go ahead.
Thank you, Christian. Very sound research, not slow, very sound. So thank you for inspiring us with all the work you do. I have a question. So these RNA binding proteins that interact with the RISC complex, they can have alternative localizations like you've shown in the nucleus, in stress granules, other membraneless organelles, and also in the mitochondria, such as too. So when you modulated autophagy, did you look at the distribution of MEX3P? Maybe it's not just nuclear, but in the other organelles, or?
We just, you know, I mean, this is a very good question, and that's something probably also to be addressed in the future. We just concentrated on nucleus versus cytoplasm, so we didn't go into further subcellular or sub-compartment specificity of MEX3A, but that's certainly something that can be done. In particular, also, I think we need to know more about, you know, where exactly this sorting occurs, because you've seen in the images also that the 3P strand goes somewhere completely different, probably more being sorted for microparticle extrusion, yeah, and so I think that the position, the exact position where MEX3A comes into the game, you know, would be very important to determine at this level.
Yeah, because I always wonder what AGO2 is doing in the mitochondria. So I've been thinking whether this distribution, when autophagy is disrupted, is to an elsewhere location. Another thing I was wondering is, you know, MEX3P knockouts, they seem to be progressing atherosclerosis.
Yes.
Do you know what would happen when you try to stabilize the plaque or regress the plaque? Would MEX3P be functional there?
We haven't looked at that yet, no. But I mean, what we've done is we've generated an adenovirus for overexpressing MEX3A in sort of carotid artery segments, for instance. So that's what we're currently doing, just as a paradigm of modulating the pathway. But we haven't looked at basically the kinetics over time and then emerging plaque or plaque evolution, or where exactly MEX3A basically behaves. But you know, I assume that because it's, you know, we've been looking at this function in endothelial cells, that also throughout the evolution of plaque, and also, you know, the flow sensitivity of basically the arteriogenesis, that it's probably still doing the same thing in earlier versus later plaques. That's at least what I would assume. Yeah.
Thank you so much.
Ada, go ahead.
Hi, can you hear me?
Yep.
Yeah.
Okay. Yeah, I think that was really interesting. Thank you for that. I was wondering if you looked at the, you know, endothelial to mesenchymal transition in those, you know, that was supposed to be like a pathway that exacerbates atherosclerosis. So.
No, you're raising all the right questions. You know, there's only so much we've been able to do. That's absolutely a great point. Yeah, so I think that should be looked at, definitely.
Thanks.
I have another question, actually, Christian. I'm wondering, I don't remember, or maybe if you said it, I missed it as well, what the targets of 5P are, and whether or not that changes sort of, you know, the effect of, you know, 5P being sort of busy in this function. Does that sort of take it away from doing its normal thing?
Yeah. So basically, the prime targets that we identified in endothelial cells were FABB6 and DLK1. DLK1 was basically the functionally most relevant target, which is a soluble inhibitor of Notch1. I mean, as to your point, I mean, we looked obviously at target availability also, because we thought maybe basically that's some explanation, you know, for the translocation, or for basically the change in in the presence, but that was not affected. So that
Okay.
was not explaining this. Yeah.
So the targets aren't acting as inhibitors of this process, basically. Okay, interesting. Yeah. Okay, do we have other questions? Looks good. I think we are good. So thank you, Christian. Thank you, everybody. And there's a Metab trainees seminar afterward, meeting afterwards, right? So
Yeah.
Yep.
those who are part of it can stay.